Hello, my name is Carla Bolaño Diaz. I am an adult neurologist and clinical research associate working at the John Walton Muscular Dystrophy Research Center. Today, I will be sharing with you one of our latest results on how MRI can improve patient care and clinical trial design. In this project in particular, we looked at how fat progresses over time and how this can be measured using MRI. So first of all, what is limb-girdle muscular dystrophy R2? This is an inherited muscle wasting disease produced by mutations or variants in the DYSF gene. This gene holds information that encodes a protein called dysferlin. Dysferlin is expressed in several tissues. However, it is key in the structure and function of skeletal muscle. It is localized to the membrane of the muscle and participates in repairing membrane after damage, which is a process that naturally happens every time we exert strain on a particular muscle, for example, when walking, running, or lifting weights. When dysferlin is absent or malfunctioning, the repair mechanism becomes faulty, and muscles are damaged and progressively replaced by fat and connective tissue over time. So, how is it that MRI can help us measure disease progression? So, a magnetic resonance or MR scanner is basically a big magnet with devices that create stimuli to modify the magnetic field that this large scanner produces. And finally, the scanner is also able to measure how different tissues respond to that modification of the magnetic field. For this study, a particular scanning protocol was used that allowed us to measure exactly the area the fat occupied, which is known as fat fraction. Fat fraction is a measure that goes from 0% in muscles that have no fat in them to 100% in muscles that are completely replaced by fat. You can see in the image here that the top two figures represent the thigh muscles, whereas the lower two represent the lower leg muscles. Muscle tissue with no fat in it is seen in a dark gray tone. You can see this, for example, in some of the lower leg muscles. Fat looks white in the images, so the whiter a muscle gets, so the lighter the shade of gray, then the more fat there is within that muscle, as you can see in the other two muscles in the thighs. This research was part of the clinical outcome study for Dysferlinopathy, the largest natural history study funded by the Jane Foundation, focusing on this type of limb girdle. 109 participants were enrolled in this sub-study, and they were scanned yearly for 3 years in 7 participating centers across the globe. We arrived to some interesting conclusions on the process of progressive muscle replacement by fat. On the one hand, in initial disease stages, lower leg muscles tended to have higher levels of fat, but later in life, thigh muscles progressively showed higher fat levels as their rate of progression increased. On the other hand, despite each muscle having its own individual trajectory, it looks as if muscles that participate in certain joint movements progress similarly over time, as you can see in the animated figure below. We also discovered that females show on average higher levels of fat in the muscles below the knees, specifically in those used to stand on tiptoes. The difference is more evident between the 3rd and 5th decade of life. People with younger age at onset of symptoms seem to have higher levels of fat than those with later onset. But at the same time, no clear link was identified between exercise levels as teenagers and fat replacement trajectory. The key question here is, how is this beneficial for the patient community? So firstly, this is more proof on the positive impact MRI can have on the study of inherited muscle conditions and how we are able to track muscle loss earlier and more precisely. Secondly, the more we understand disease progression, then the better we can advise people living with this disease and plan their care ahead of time. It reinforces the fact that progression is not 100% homogeneous among patients, and that a patient-tailored care approach is non-negotiable. Finally, these results can help us improve the design of clinical trials depending on the population that will be included and the final outcome. Thank you very much for your attention. I hope you enjoyed this video, and if you want to read more about this project and access the whole manuscript, please go ahead and scan the QR code you're seeing on this slide. It will take you directly to it. Thank you very much.